Injury from ischemic stroke is the result of a complex series of cellular metabolic events that occur rapidly after the interruption of nutrient blood flow to the region of the brain. The duration, severity, and location of the focal cerebral ischemia determine the extent of brain function, thus the severity of the stroke. Blood flow to the region on the left is sometimes interrupted due to a thrombus or an embolus. The lack of blood flow results in severe damage to the brain tissue. The infarcted tissue causes fluids to accumulate and result in swelling. The center of the brain is shifted to the right from the left. The rigid container of the cranium allows limited room for expansion, and any condition that causes an increase in volume of one or more of the structures within this vault will cause an increase in intracranial pressure, or ICP, thereby compressing other compartments. As the pressure increases, the brain shifts or is distorted, compressing neurons, nerve tracts, and cerebral arteries. A sustained increase in pressure causes persistent ischemia and irreversible damage to brain cells and possibly death. A neuron consists of a cell body which contains a nucleus and one or more extensions protruding from the cell body. Dendrites receive nerve impulses from other neurons or from sensory receptors. The axon carries the nerve impulses away from the cell body to another neuron or to an effector organ, such as a muscle. A stimulus affects the axon by changing the permeability of the axon to positive ions. The influx of positive ions reduces the electrical potential across one segment of the membrane, depolarization. The change in electrical potential in the first part of the axon triggers a change in electrical potential in the adjacent segment of the axon, such as the impulse travels along the axon to a self-generating chain reaction. In most synapses, arrival of the impulse at the presynaptic terminal leads to release of a neurotransmitter, which crosses the synapse to interact with receptors on the membrane of the postsynaptic cell. This interaction opens an ion-specific channel in the postsynaptic membrane, changing the membrane's permeability for positive ions. The transient change in voltage induced by the action potential is determined by the concentration of ions on either side of the cell membrane. Maintaining these ionic gradients is an energy-consuming process that requires a constant supply of glucose and oxygen to the neurons. Lack of glucose and oxygen deplete cellular energy stores required to maintain electrical potentials and ion gradients. In ischemic brain tissue, the membrane that surrounds the affected neuron becomes leaky, and the cell loses potassium and adenosine triphosphate, ATP, the tissue's medium for energy exchange. Energy failure is not the immediate cause of cell death, however, since all brain cells tolerate a loss of ATP for several minutes. In humans, it appears that 5 to 10 minutes of complete occlusion is required for irreversible brain damage. In actuality, most strokes do not involve a complete occlusion of blood flow, but even a partial occlusion is allowed to continue for a sufficient time may produce irreversible brain damage. Once blood flow to the cerebral neurons diminishes, one or more branching mechanisms may independently lead to brain cell death. These mechanisms may involve deterioration of ion gradients or the effects of anaerobic metabolism. With respect to the latter, anaerobic glycolytic pathways are utilized in the affected region to compensate for the loss of oxygen and provide a source of energy. However, this produces damaging byproducts, including lactic acid and hydrogen ions, which accumulate in the tissue in proportion to carbohydrate stores present in the outset of ischemia. Toxicity of hydrogen ions, especially their ability to facilitate ferrous non-mediated radical-free mechanisms, appears to irreversibly affect neuronal integrity. Inadequate energy supplies leads to deterioration of ion gradients. Anoxic depolarization equilibration of intracellular and extracellular ions, causes potassium to leave the cell and sodium chloride and calcium ions to enter. It also stimulates the massive release of the amino acid glutamate and aspartate, excitatory neurotransmitters in the brain. Glutamate further activates sodium and calcium ion channels in the neuron membrane. As sodium and calcium ions rapidly accumulate within the cells, accompanied by an inflow of water, cytotoxic edema causes rapid swelling of neurons and glia. Activation of calcium channels results in further influx of calcium into the cell. One of the most intensely studied calcium channels is the N-methyl D-aspartate channel, NMDA. TPA is classified as a serine protease, enzymes that cleave peptide bonds in proteins, and thus one of the essential components of the dissolution of blood clots. Its primary function includes catalyzing the conversion of plasminogen to plasmin, the primary enzyme involved in dissolving blood clots. Recumbent biotechnology has allowed TPA to be manufactured in labs. These synthetic products are called recombinant tissue plasminogen activator, RTPA. Examples of these drugs include alteplase, retaplase, and tenecteplase. These drugs have undergone various modifications to amplify their pharmacokinetic and pharmacodynamic properties, especially to prolong their short half-life in the circulation and further increase their fibrin specificity to prevent an unwanted fibrinolytic state. TPA is a thrombolytic. It breaks up blood clots formed by the aggregation of activated platelets into fibrin meshes by activating plasminogen. More specifically, it cleaves the zymogen plasminogen at its AR561 VAL562 peptide bond to form the serine protease plasmin. Plasmin is endogenous fibrinolytic enzyme, breaks the cross-links between fibrin molecules, which are the structural support of the blood clot, and its activity is extremely short-lived. This is because alpha-2 antiplasmin, an abundant inhibitor of plasmin, quickly inactivates it and restricts the action of the plasmin. TPA attaches to the fibrin on the blood clot. It activates the fibrin-bound plasminogen. Plasmin is subsequently cleaved from the plasminogen affiliated with the fibrin. The molecules of fibrin are broken up by the plasmin and the clot dissolves. Plasminogen activator inhibitor eventually terminates the catalytic activity of TPA by binding to it, and this inactive complex is removed from the circulation by the liver, more specifically via the scavenger receptor LDL receptor-related protein 1. In the nervous system, a neuronal specific inhibitor of TPA, neuroserpin, acts similarly to PAI1, and the inactive TPA neuroserpin complexes are internalized by the LRRP1 for removal from circulation.